Okay, chapter 30, cytogenetics. So we'll talk about what chromosomes are all about, what the structure is, why we do it, how we identify different parts of the chromosome, what is actually done to prepare the specimen to get the chromosomes and how we analyze it, what's the actual name of the stuff, some of the chromosome abnormalities, certain other type of leukemia, cytogenetics, and again, here comes that chromosome microarray analysis that we're talking about. So, chromosomes, that's basically where we inherit stuff from our parents. This is where DNA is located. So the chromosomes, we break them either in structural or some numerical thing to see if we either lose or gain or transform or translate, a piece of a chromosome, a part of a chromosome, or the entire chromosome. Because anytime the chromosomes are altered or changed or anything like that, any kind of disruption of any kind can cause consequences. We can cause, and we're just going to focus just on the leukemia kind of stuff. All right, so to look at chromosomes, to analyze them, we actually can spread them up into a map, which you'll see here in a few seconds, and it's called a karyotype. So the karyotype is where we actually look at the different types of chromosomes. And from that, you can determine, if there's a mental retardation, anything that happened at all with the chromosomes, cancer, genetic risk, and you can see the rest of them that are there that we can analyze from the chromosomes. So look, this is a karyotype. That's the best way, just look at it. So they match up, remember we have 23 chromosomes, 22 are autosomal. This 23rd is a sexual, sex-linked one. That's one where we have the X or the Y, and you can see the pictures of those. So they look at these chromosomes, and you can see there's, some are darker, lighter. That's the different sections of the chromosome. They call that, in this particular case, a G banding is what's actually done in this particular one. But they match them up, 1, 2 through, all the way through 23. And they look at it and they compare them and, to make sure there's any kinds of problems. So where does this chromosome structure come from? So you can see at the very bottom is DNA helix, and then they make a double helix, and then it, which contains all the genes. So then they bind tight together to make a double helix, which then they form fibers, where it's packed into nucleosomes, and then they're extended and wrapped and coiled and condensed to get to the chromosome. I mean, you can see how it goes from one phase to the last. So the metaphase, which is the top one, that's where we actually can see, more individual chromosomes to get more information. So you're talking about millions and millions and millions of strands of DNA in a single chromosome. Remember, we got 23. And of course, it's one from each parent. So this is meiosis, not mitosis, but meiosis. So the metaphase is where we look at the individual chromosomes, because that's where all the folding and all that goes inside the chromosome itself. So the chromosome numbers, somatic is 46 chromosomes totally, one from each parent. That's a haploid number, so that would be 23 chromosomes, so one from mom, one from dad. So when we look at the chromosomes for identification, we actually have them, different techniques that are done. They call them banding techniques. G is the most common one that's used, particularly around us. But there's 7 different groups, A through G. And what they're looking for is a metacentric, the submetacentric, and an acrocentric. So you can see there's different parts that they're looking inside of the chromosome itself, to band it, in other words, to identify what we're looking for. There's a centromere in the middle. So, what kind of specimens do you deal with? Well, bone marrow is the best because we have higher mitotic rate, because remember, you got the blast, stem cell, all that junk is in there. You can use peripheral blood, but bone marrow seems to be the best. So when they get the bone marrow cells, what they'll do is they stimulate the division so it's faster now. And then when it gets to the metaphase, that's where they actually stop it. Because once we have chromosomes, this mitosis, that's going on. So they disrupt the mitotic spindle, so it can't, for the attachment. So what they'll do is they put it in a hypotonic, solution, and that makes the chromosomes like split apart. And then they fix it, and then they put them on a slide and expose them to heat, and then they get pictures of it. People that do this is amazing. I would love to see it. So, like I said, the chromosome banding, we get Q banding, G banding, GTG banding, C banding, Agnor banding. G banding is the most common that we use in our particular area. So how to come up with, when you look at cytogenetic abnormalities, they got the Qs and the Ps and all this junk in it. The short arm, which I'll show you a picture in a second here, and this is all the way back to 1971. The short arm is considered the P, the long arm is considered the Q, and then they got the different number of bands that's in the chromosome, which you'll see in the picture. So he said there's 1 to 22 is somatic, and the 23rd is the sex chromosome. So let's look at a picture. So P, here in the center is the centromere. So P is a short, Q is a long, and you can look and see the different numbers that come with it. Now, I don't know how they do it. I really don't know, but it's pretty amazing on how they do this kind of stuff. So when you look at it here, so chromosome analysis, one of the biggest things we do is called FISH. Now, when you break FISH down, that stands for fluorescence in situ or in situ hybridization. So what they do is they get DNA, okay, and it's a probe, it's labeled with something. Usually, in this particular case of FISH, it's labeled with a fluorescent dye. All right, so what they'll do is they get the chromosomes. On a slide, they'll have the chromosomes laying there. They then apply the labeled chromosome that is bound to it, that's got a fluorescent marker to it, and they put them on a slide. So the slide will contain the patient's fluorescence that has been matched to a labeled probe to a certain area of the chromosome. So what they'll do is they'll denature it, hybridize it overnight, wash it, rinse, wash, rinse, wash, rinse, and basically you look at it under a fluorescent scope. So let's just look at a picture of it. It makes more sense. So when you look at a picture of it, you can hardly see it, but there are some chromosomes in the back. But if we have this piece of DNA that has been attached with a fluorescent probe, a fluorescent label to it, and they have that genetic abnormality that matches that probe, it will then be fluoresced. Like it's showing a red pair here and a green pair here. This is called a BCR-ABL. It's a 9 and 22 chromosome is what we're looking at. And then we'll talk about that particular one later, but this is just an example. So we can do DNA and RNA probes using this FISH techniques to help us identify certain chromosomes, which is great. Another part of where the WHO classifications come from. Flow cytometry with immunophenotyping and cytogenetics. This is it. So for the chromosome abnormalities, what we look for is structural problems, and these structural problems can either be an inversion or a deletion. They can be multiple chromosomes, it can be rings, translocations, duplications, all kinds of things that can happen with the chromosome abnormalities. So what are the clinical applications of this? Well, it helps us to identify exactly what type of abnormality people have. I mean, if you look, in your textbook, you can see, they got pictures of all kinds of FISH things showing you different abnormalities. And it's not important that you know all the different abnormalities yet. We'll get more into that when we get to leukemias and stuff like that. But, as structural things that have happened, things that have changed. So from that, dealing with just hematological stuff, leukemias, we can determine if it's lymphoid or myeloid, and you can see the different categories we're looking for. ALL, AML, CLL, CML. And this is where the WHO classification comes into play. Do we get a better diagnosis as to which type of leukemia? Remember, the FAB classification is just a morphological identification, so it's not really cytogenetics per se yet. All right, so for instance, CML, chronic myeloid or myelogenous or myelocytic leukemia, that was the first one we had identified that had a chromosome abnormality. So that's where we had a BCR-ABL, the little green and red we looked at a few minutes ago. That's a translocation of the 9 and 22 chromosomes. And the name for that is called Philadelphia chromosome. So it causes a BCR-ABL fusion. In other words, the cells don't die, basically, is what's happening. They just live forever. That's why the counts climb. But anyway, so we detect that using FISH, which we just saw there. Now, what's nice about identifying that is that we have a medication just for that now that will stop that BCR-ABL chromosome abnormality. And because of that, treatment is very good, can work well, and then we can monitor it by doing FISH technology to determine if they have that Philadelphia chromosome present or not. Here you can see some acute leukemias. Again, Philadelphia chromosome can be present. That's acute lymphocytic, acute myelocytic, and then there's a whole bunch of translocations here, and that tells you the location, or it is translocation of 4 and 11 on Q21 and Q23. You know, another example, you know, when you're talking about solid tumors, is HER2, which is human epidermal, something I can't remember what it stands for now, but that indicates a certain kind of breast cancer. So watch commercials, and you'll see when there's this HER2, you'll hear them talk about it. And that's good for determining prognosis and tells you exactly what kind of treatment, you know, therapy that can be used. So that works really well. Microarrays that can be done, same that we were just talking about with the molecular thing. We can look for multiple chromosome abnormalities here, you know, for cytogenetics. Instead of just molecular, we can look for chromosome using the same kind of basic technology that we were just talking about. So, cytogenetics we went through pretty quick, but, you know, it's looking at the different types of chromosome abnormalities. You know, we don't really need to draw a label of DNA molecule. We're not getting into that. But, you know, what does FISH stands for? What is the P and Q? You know, how many chromosomes do we have? What abnormalities do we have? What is a translocation? What is a deletion? What is an insertion? What is a point mutation? I mean, what is all that stuff? That's important to know. You can look at a review and the summary that's in the back of the chapter and some of the review questions because the answers are in the back. But again, the chapter review is the best thing to help us study through. I'll be making a recording of that in a little bit, so you can use that to help study, and we'll be talking about it in lab also. If you have any questions, make sure you contact me. Thank you.